Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled 3 Key Trends Transforming the Future of Rare Disease Clinical Development. My name is Mohammed Ali and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the Comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank MedPace, who developed the content for this presentation. MedPace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. MedPace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach that leverages local regulatory and deep therapeutic expertise across all major areas. Now, I'd like to introduce our speakers for today's event. Dr. Anne Wolfrey is board certified in internal medicine and pediatric hematology and oncology and provides clinical and scientific expertise in cellular therapies, transplantation, immunology, hematology, oncology, and gene therapy. Dr. Wolfrey joined MedPace 9 years ago after a 25-year career in translational and clinical research in cell and gene therapies at the Fred Hutchinson Cancer Research Center / University of Washington. In addition to a role as the medical director of the Unrelated Donor Program at FHCRC Seattle Cancer Care Alliance, she was principal investigator on numerous clinical trials and has more than 150 scientific publications in the field of stem cell transplantation, transplant immunology, HIV, and autoimmune diseases. Tanya Koval-Valenko has over 25 years of international experience in the drug development of small molecules, biologics, and combination products. Her expertise spans all phases of clinical research through authorization and post-approval lifecycle management. Having worked extensively in Canada and Switzerland, Tanya has served as a principal interface for interactions with major regulatory agencies, including the FDA, EMA Health Canada, and various national competent authorities in Europe. She possesses deep expertise in designing and executing effective global regulatory strategies, managing pre-submission meetings, and leading complex negotiations with agencies. Jackie Widmer has 20 years of global industry experience leading phase 1 through 4 studies across multiple therapeutic areas, including over 13 years of MedPace global project management and progressive leadership roles. She has over 5 years of pharmaceutical experience leading global rare disease and gene therapy development programs. And now, without further ado, I'd like to hand the mic over to our first speaker. You may begin when ready.
Okay, thank you. As Rare Disease Day approaches tomorrow, we take this opportunity to recognize the global impact of rare diseases and the urgent need to accelerate the development of safe therapies to improve the lives of patients. Rare disease clinical development is undergoing rapid transformation. It's driven by scientific innovation, evolving regulatory frameworks, and the need for more flexible, patient-centric trial designs. As the rare disease landscape is transforming, these clinical trials are becoming more complex and require CRO partner and global resources, extensive experience, and strong collaboration. In today's webinar, MedPace leaders will explore 3 trends transforming the future of rare disease clinical development, including advantages and considerations of platform trials for rare diseases, 3 major shifts in regulatory frameworks, the FDA's new evidence principles, China's aggressive 30-day trial entry, and the specialized strategies required for the ultra-rare. And ex vivo gene therapy as an emerging technology for treatment of inherited diseases of the blood or immune system. Next slide. To begin, I'd like to introduce platform trials and the value that they bring to rapid and efficient development of therapeutics in rare diseases. Next slide. A platform trial can be designed to study multiple targeted therapies in the context of a rare disease in a perpetual manner, with therapies allowed to enter or leave the platform. When multiple treatment candidates are available, this can lead to quick scientific and therapeutic discoveries. Platform trials have important potential advantages in creating an efficient trial infrastructure that can help address important clinical questions as the evidence evolves, particularly in rare diseases. The master protocol lays the foundation and dictates the overall study design and analysis. It describes the overall common inclusion and exclusion criteria, in which can be shared across homogeneous patient populations, as well as a common schedule of assessments that will be collected across all sub-protocols, or otherwise known as intervention-specific appendices, or ISAs for short. Within the platform, each sub-protocol, or ISA, will focus on specific investigational product. Platform studies can go on perpetually and can maintain flexibility to run multiple ISAs at one time or in sequential order. Next slide. Platform studies are gaining momentum in rare disease research because of the scientific and operational efficiencies they create. These efficiencies not only accelerate therapeutic discovery, but also streamline trial execution in populations where time and patience are limited. Rather than building a soccer stadium to host a single match, a platform study is like building a stadium to host an entire tournament. Once the infrastructure is in place, multiple interventions can be evaluated within the same framework, reducing redundancy and maximizing resources. Under a platform model, all study activities are governed by the single master protocol that defines the overarching design and conduct of the trial. Sites are selected with the intention of participating across all ISAs. After initial startup and activation under the master protocol, subsequent ISAs are typically implemented through regulatory and contractual amendments, allowing new interventions to be launched more efficiently with the existing structure. New therapies can be introduced either simultaneously or sequentially in the same patient population. In many platform designs, placebo data can be shared across all ISAs, reducing the number of patients assigned to placebo and increasing the likelihood that participants receive active treatment, an especially meaningful advantage in rare diseases. Additionally, platform studies enable the collection of robust natural history data within a unified database using standardized assessments over time. This consistency enhances data quality and provides valuable long-term insights into disease progression, which is often limited in rare disease progression. By creating an adaptable and enduring infrastructure, platform trials allow sponsors to rapidly introduce and evaluate new interventions. Ultimately, this model accelerates the development of novel therapies and brings meaningful advances to rare disease communities more efficiently than traditional standalone trials. Next slide. MedPace has extensive experience establishing and executing platform studies. To provide a high-level overview, I'll walk through an example of how a platform study is structured and operationalized. At the core of any platform study, as mentioned, we have the master protocol. And again, this serves as the overarching governing document. The master protocol defines the overall study design, targeted patient population, primary and secondary objectives, and the common assessments conducted across all interventions within the platform. It also guides country and site selection, typically with the intention that sites participating in the platform may engage in multiple ISAs. With that said, the participation in the platform does not obligate a site to participate in every ISA. Flexibility is built in to accommodate site capabilities and regional considerations. Operational consistency across the platform is critical. Key study systems such as IRT, EDC, and central laboratory services are generally established at the master level and leveraged across all ISAs. In partnership with sponsors, MedPace develops master-level operational plans and defines governance, processes, and collaboration models. Depending on the complexity of a platform, ISA-specific operational plans may be developed as well. Certain documents and processes remain ISA-specific. For example, one would expect investigational product-related materials such as an investigator brochure, pharmacy manual, and clinical supplies are typically intervention-specific. Likewise, certain assessments or laboratory requirements may only that apply only to individual ISAs rather than the entire platform. Safety reporting and oversight are generally governed at the ISA level to ensure appropriate regulatory compliance for each investigational product. While analyses are conducted separately for each ISA, master-level statistical analysis plans may also evaluate pooled or overarching platform data. Clinical study reports are typically generated for each ISA, with potential for integrated reporting at the platform level where appropriate. One of the key advantages of the MedPace model is continuity. A single, dedicated study team that supports the platform from initiation through completion, ensuring consistency across ISAs and long-term partnership with sponsors. Regulatory strategy is critical to the consideration in platform trials, particularly when multiple interventions are under investigational simultaneously. Activities such as investigator brochure updates, protocol amendments, and annual DSUR submissions must be strategically coordinated to maintain global regulatory compliance and efficiency. Following initial site activation under the master protocol, additional ISAs can often be implemented through streamlined processes similar to protocol or contract amendments. This significantly accelerates the activation of new interventions within the established infrastructure. Oversight and performance management are maintained at both master and ISA levels. MedPace collaborates with sponsors to establish KPIs and metrics that ensure robust governance across the platform. Operational efficiencies are further enhanced through consistent monitoring, enabling monitors to review multiple ISAs during a single visit. TMF management, training, and quality oversight are likewise structured at both levels to ensure compliance and consistency. Once the foundational systems, procedures, and governance structures are established, incorporating new interventions becomes significantly more efficient, facilitating the rapid advancement of promising therapeutics, particularly in rare disease populations. Platform studies represent a transformative approach to drug development in rare diseases by increasing operational efficiency, reducing redundancy, and accelerating timelines. I've introduced the new trend of platform studies and how they can enhance drug development in the rare disease population. In the next session, my colleague Tanya will discuss new global regulatory frameworks transforming the rare disease landscape. Next slide.
Thank you, Jackie. Good morning, good afternoon, everyone. As we observe Rare Disease Day, we find ourselves at a pivotal moment. Today, I will cover three major shifts in global regulatory affairs drug frameworks. To begin with, we will review how the FDA evaluates therapies for ultra-rare diseases, new rare disease evidence principles. Next, I will move on to fast-track IND process in China, which is China's aggressive 30-day trial entry. And then I will go on to highlight the specialized strategies required for the ultra-rare, where the global patient population may be fewer than 1,000 people. So if we go to the next slide, please. In September 2025, the FDA introduced the Rare Disease Evidence Principles, RDEP. This is not just a new guidance, it's a commitment to a common sense approach for ultra-rare diseases with genetic defects. It's a joint CDER and CBER process that aims to provide speed and predictability. Under rare disease evidence principles, the FDA now accepts one adequate trial plus robust confirmatory evidence. This evidence can be pharmacodynamic data, mechanistic or biomarker evidence, natural history studies, registries, or even case reports forms from expanded access programs. To qualify for RDEP, a therapy must meet specific criteria. Eligibility is strict, targeting diseases with a known genetic driver and fewer than 1,000 patients in the US. This is below the standard orphan drug limit, which is fewer than 200,000 patients. Next, it should be severe progressive condition that causes rapid disability or death. And finally, there are no existing disease-altering therapies. Next slide, please. I just want to bring to your attention that RDEP process is separate and independent from the established FDA orphan drug designation. Request for RDEP review must be filed prior to the launch of a pivotal trial as part of a formal meeting request. To summarize, RDEP is not a lowering of standards, it's a widening of lenses. For years, the two-trial rule was the death of rare disease programs. RDEP now provides the roadmap for what constitutes scientific rigor when a second trial is ethically or logistically impossible. Next, please. While the US is innovating on approval evidence, China has revolutionized trial entry. In October last year, the National Medical Product Administration in China formalized the 30-day expedited IND pathway for innovative drugs. Next slide, please. If your drug targets an orphan indication on China national list, the sponsor is encouraged to take advantage of a 30-day review pathway, so you get your decision within 30 working days instead of 60 working days. But here is the catch. The 12-week rule is applied. You must legally commit to screening your first Chinese subject within 12 weeks after IND approval. The introduction of the 30-day expedited IND pathway has massive implications for global multi-regional clinical trials. By using this track, the sponsors developing therapies for rare and ultra-rare diseases can effectively sync your China phase 2 or 3 start with the US and EU timelines. And it also encourages early involvement of Chinese principal investigators in phase 3 studies. Next, please. In addition, China has finally addressed the intellectual property gap for orphan drugs. The new policy that will take effect in May this year will formalize market exclusivity period for rare disease therapies, preventing follow-on competitors from entering the market. This is a massive win for asset valuation. This will be in addition to existing 6-year data protection period from the date of China marketing authorization, which prevents competitors from using originators' undisclosed clinical trial data to gain approval for generic or biosimilar versions of a new drug. When you combine this with the 3% reduced VAT for imported orphan drugs and accelerated inclusion in the national reimbursement drug list for approved orphan product, the path for a profitable high-speed launch is apparent that it has ever been. The Chinese regulatory agency is essentially rolling out the red carpet for rare disease innovators. Following implementation of these two regulations, China is no longer a phase 3 after third, with 7 years of exclusivity, a 30-day entry clock, and a government-driven mandate to solve unmet rare disease needs. China is now a strategic pillar for any orphan drug development program. Next slide, please. Finally, let's look at the ultra-rare frontier. As we observed in 2026, the regulatory approach for ultra-rare diseases has moved toward totality of evidence models. Next slide, please. In late 2025, the FDA also introduced the new concept, plausible mechanism pathway. That is designed to accelerate approval for highly customized or individualized therapies targeted at very small scale of patients, particularly for ultra-rare, severe, or genetic diseases. The pathway will allow us to lean on evidence of a plausible biological mechanism of action and a well-defined molecular target like a protein expression or genetic correction. The pathway will heavily rely on the expanded access investigational new drug process as well, and a single patient IND. Although safety and efficacy data from a single patient IND have not historically served as the basis for approval, the plausible mechanism pathway would allow even anecdotal data for individualized therapies to be used as a potential sources of evidence. If consecutive patients show successful results, the FDA may grant marketing approval even without standard large-scale randomized trials. In the same time, post-marketing activities will require a stringent ongoing data collection to confirm efficacy, safety, and lack of off-target effects. Next slide, please. So in the landscape of ultra-rare diseases, the traditional fixed trial design fails because there simply are not enough patients to reach statistical significance. Two powerful tools, Bayesian adaptive designs and the use of external control arms, can now solve this. Utilization of smaller high-frequency data collection points to maximize statistical power in tiny cohorts and leveraging natural history studies and real-world evidence to eliminate placebo arms can accelerate regulatory approval and reduce the number of participants required for rare disease studies. Crucially, last and not least, the new Joint Artificial Intelligence Principles released last month by the FDA and EMA now provide a clear roadmap for using artificial intelligence to generate evidence for supporting marketing authorization. This is a very important step to overcome a small patient population size by creating synthetic control arms and identifying eligible patients from global electronic health record databases. Next slide, please. Today, the roadmap for ultra-rare success is built on three main pillars. First, a global protocol or master protocol that pivots to decentralized trial model to reach geographically dispersed ultra-rare populations. It also overcomes the challenges of small patient populations and the high cost of traditional drug development, while ensuring that your data will satisfy the FDA's substantial evidence and the EMA's significant benefit simultaneously. China is now a must include for ultra-rare development. Second, use of innovative trial designs such as adaptive trial designs, micro trials, and patients acting at their own controls. These are no longer experimental trials, but necessary responses to the ethical and operational realities of ultra-rare diseases. Real-world evidence and natural history data should be considered as external control arms to meet standards where traditional trials are unfeasible. Third, don't forget CMC, chemistry, manufacturing, and controls. For ultra-rare, your manufacturing consistency is often scrutinized as much as your clinical data. So to recap, we can now observe the shift toward regulatory agility. Regulators are increasingly accepting a totality of evidence in addition to well-controlled studies. My final recommendation to you is this. Don't wait to phase 3 to talk to the FDA or Chinese competent authority or any other important regulatory authority in the world. Use this new 30-day expedited pathway, rare disease evidence principle pathway to build a global program for your patients today. Your robust regulatory strategy and operational readiness is now your greatest asset. This is the end of my presentation, and thank you for your attention. Now I hand it over to Anne.
Thank you, Jackie and Tanya, for your very interesting presentations. The scientific advances in the field of gene therapy are happening quickly, and your information will help spur clinical development for rare diseases. In this part of the webinar, I'll present information about an important emerging technology called ex vivo gene therapy. Next slide. In order to understand how to cure certain inherited diseases of the blood or immune system, we need to understand how blood and immune cells develop. All blood and immune cells originate from a primitive cell termed the hematopoietic stem cell, or HSC, that resides within the bone marrow. The blood and immune systems are maintained because the stem cell can divide and continuously make new stem cells. Under certain conditions, the stem cell will start the process of differentiation. This process produces the different component cells of the blood system, such as platelets, red blood cells, and white blood cells, including myeloid cells, such as neutrophils, and lymphoid cells, such as lymphocytes. Once the differentiation process starts, the stem cell is committed and cannot self-renew. The mature differentiated cells have short lifespans. Thus, a permanent correction of an inherited disease requires replacement or correction of the mutated stem cells. Replacement of the stem cells done through bone marrow transplantation, whereas ex vivo gene therapy is the procedure used to correct the genetic mutation within the stem cell. Shown on the right side are the indications for ex vivo gene therapy. This includes genetic disorders of hematopoietic stem cells, red blood cells, myeloid cells, and lymphocytes. Diseases that arise from the common hematopoietic stem cell can be corrected using gene editing or gene transfer techniques. Ex vivo gene correction employs three main platforms depending on the characteristics of the genetic mutation. Each has distinct mechanisms, advantages, and technical challenge. Gene augmentation is pictured on the left, involves the delivery of a normal gene into the stem cell. While the abnormal gene remains, the idea is that enough of the normal protein is produced so that the cell can function correctly. Candidate diseases that could be cured by addition of a normal gene include certain types of immunodeficiency syndromes, as well as syndromes that have more than one affected cell type, such as the combined platelet and immune cell deficiency seen in Wiskott-Aldrich system. On the right is pictured the platform of gene disruption, which involves the delivery of an editing machinery, such as a CRISPR-Cas9, that cuts out part of the mutated gene, thus blocking the mutated gene from production of the abnormal protein. Candidate diseases that could be ameliorated by disruption of a dysfunctional or unwanted protein include the hemoglobinopathies, such as sickle cell disease and beta thalassemia, or potentially HIV. Genome editing is shown in the middle, and that combines both gene disruption to remove the mutated area within the gene and procedure to replace the mutated area with the correct gene sequence that results in a normal protein. Candidate diseases that could be cured by genome editing include sickle cell disease and certain immunodeficiency diseases such as chronic granulomatous disease. Selection of the appropriate platform depends on the characteristics of the specific mutation. Is there a single genotype, or are there multiple genotypes that are associated with the disease? What is the consequence of the mutation? Would there be a negative or interfering effect of the uncorrected protein, or is that protein absent? What is the required level of protein expression for correction of symptoms? Certain diseases require low engraftment of gene-corrected cells, such as primary immunodeficiencies, whereas other diseases require high engraftment, such as beta thalassemia and lysosomal storage disorders. Do the gene-corrected cells have survival advantage? And is there a requirement for tissue specificity of the gene expression? By considering these requirements for correction of an individual disease, ex vivo gene therapy platforms have achieved remarkable clinical success for a number of inherited hematologic and immunologic diseases. The list of products that have achieved regulatory approval thus far are shown here, and many more studies are underway. Next slide. The technical procedure required to produce a gene-modified stem cell product is quite complex, and the technical procedure is only a small part of the overall ex vivo gene therapy. The technical workflow is illustrated by the period of time encompassed by the green arrows on the figure. To generate the gene-modified cell product, the stem cells must first be collected from the patient. Next, one of the platforms is used to genetically modify the stem cells, after which the product is frozen. Once the product has been manufactured, the patient can be treated. They must first receive conditioning chemotherapy to make room for the hematopoietic stem cells in the marrow, and then the product can be infused intravenously into the patient. However, as you can see, when we bring up this next part of the slide, a great deal of trial work surrounds the technical process of delivering gene-modified cells. This includes the recruitment and referral of rare disease patients, screening and selection of the eligible patient, oversight from a transplant team for collection of the stem cells, oversight from the transplant team for transplant procedure and the early recovery period, and medical care throughout by the disease specialist before and after gene therapy procedure. As well, importantly, collection of the data to support the endpoints of the trial and long-term monitoring for late effects. The complexity of this type of protocol demands that a CRO will have the specialized knowledge, the clinical experience, and strong relationships with sites that are capable of conducting the trial. Next slide. Let's look deeper at some of the challenges for a clinical trial to be successful. Oftentimes, these patients are extremely rare. Given the risks of the procedure, patients should have few, if any, curative options. However, patients with a chronic disease may be reluctant to take on the risks of an intensive therapy, even if their disease will ultimately shorten their lifespan. Patients who are willing to enroll in a gene therapy trial may actually not be fit for a myeloablative conditioning due to complications of their disease. And once eligible, patients may not be willing to take on the time commitment, which generally entails months of treatment and monitoring at a specialized stem cell transplant center. It's important to have a robust recruitment and retention group in the CRO to help find these difficult, rare patients to identify. Once a patient arrives at the center, there will be a screening period to determine if the patient meets eligibility requirements. Most often, the physician caring for the patient with an inherited disease will not be the same as the physician directing the procedure. Determination of fitness for myeloablative conditioning can be challenging. Patients may have infections or chronic organ dysfunction from their disease. And this field currently lacks a validated comorbidity index that can be used to rank or rate patients' fitness for the procedure, which is in contrast to our bone marrow transplant procedures, which have a comorbidity index that can predict risks for the procedure based on the patient's organ function. One issue is whether the bone marrow needs to be screened for clonal hematopoiesis. This may be indicated in selected patients. Currently, there's no consensus guidelines to determine who needs screening, and there's no standardized management if clonal hematopoiesis is detected. Infertility is a big concern, particularly for female patients, as it is a potential risk from busulfan conditioning. Fertility preservation requires time, sometimes months, and expense. Disease control must be maintained during the screening process. Certain medications used to control the disease may have a negative effect on stem cell viability and may require a washout period. Next slide. The collection of stem cells requires a command of the logistics and knowledge of site capabilities. Apheresis is the standard way to collect hematopoietic stem cells. However, there are a logistical and technical challenge. The apheresis procedure must be performed by specialized technicians, and this is particularly the case for pediatric patients. Apheresis slots are often scheduled weeks in advance, and the slot scheduling must consider the time to transport the stem cell product and avoid weekend delivery to the manufacturing site. Mobilization of hematopoietic stem cells can occur in the clinic, but that must be scheduled after the apheresis slot is confirmed, because the standard procedure using 4 days of GCSF must consider when the collection is determined to occur. In certain diseases, such as sickle cell disease, however, GCSF mobilization is contraindicated. Plerixafor is often used in both GCSF mobilized and for patients who cannot receive GCSF to help increase the number of stem cells collected. Because genetic manipulation typically requires a large number of hematopoietic stem cells, the targeted cell dose may be high. It may require more than 2 days of collection, or it may require more than one apheresis procedure to achieve that targeted cell dose. Furthermore, there's often a requirement to collect additional cells that are not genetically modified to be used as backup cells to be reinfused in case of poor hematopoietic recovery. Venous access may be an issue in patients who have a history of IV therapies. Poor venous access may necessitate placement of a temporary apheresis catheter. Catheter replacement requires scheduling with surgery or an interventionalist. Patients with sickle cell disease may need exchange transfusions before catheter placement. The apheresis technique may need to be altered in order to achieve adequate collection. For example, collection of peripheral blood stem cells from patients with sickle cell disease requires altering the level of cut through the buffy coat. These technical challenges are important to understand in order to bring a patient successfully through the apheresis collection. However, very young patients may not tolerate apheresis, and stem cells may need to be collected by a bone marrow harvest. This also requires advanced scheduling of an operating room in an experienced pediatric bone marrow transplant program. A specialized skill set is required for harvesting stem cells from pediatric patients. Large cell dose requirements for product manufacturing, generally 30 ml per kilogram, may bring the potential need for blood transfusions for these very small patients. Next slide. Once the stem cells are collected, they need to be shipped to the manufacturing facility. The time for manufacturing the final product varies, but can be several weeks. During the manufacturing period, the patient must continue to receive medical care with a disease specialist so that they remain eligible for the transplant. The logistical steps at the site include packaging the stem cells and scheduling pickup, as well as receipt and storage of the final product from the manufacturer. Chain of custody of the hematopoietic stem cell product is extremely important, as outlined here. The product must carry patient-specific identification throughout the entire process, and each step must appropriately document the chain of custody. MedPace has considerable experience in training sites in the logistics and managing the chain of custody. Next slide. Once the product has been manufactured, the patient will be scheduled to undergo the transplant procedure. These procedures are performed by physicians specialized in blood and marrow transplant. The disease specialist also plays an important consultative role, as disease-specific symptoms may need management during the long procedure. Before admission, it must be confirmed that the patient remains fit for receipt of a myeloablative conditioning regimen. Scheduling the transplant generally involves an inpatient admission for conditioning and transplant of the product. Protocols and cell therapy manuals should specify the details of the procedure. Optimal conditioning with busulfan requires dose adjustment based on the area under the curve. Institutional standard practice varies with respect to busulfan monitoring. Some sites have in-house busulfan labs, and some will need to send out blood samples. The turnaround time for receipt of busulfan levels from the outside labs may vary. Note that the starting dose of busulfan is generally higher for pediatric patients because their rate of metabolism of busulfan is higher. Once the busulfan has cleared, the stem cells can be infused. Generally, this is about 48 hours or more after the last dose of busulfan. Thawing of the cell product may occur at the bedside or in the cell therapy lab. It's important that sites have experience with the stem cell infusion apparatus, including the pumps and lines. Note that infusion reactions are common and can be minimized by pre-medications. There's also the risk of an anaphylactic reaction, so anaphylactic precautions are important. Bone marrow transplant units have standard practice for prevention of infections, but there may be disease-specific supportive care that needs to be added or included in the protocol. Note that hematopoietic recovery after myeloablative therapy and infusion of genetically modified cells generally takes longer than what we see with a typical autologous transplant procedure. Because reconstitution of hematopoiesis takes time, the ameliorating effect of the normalized immune or blood system will not be observed for many months. During that time, the patient will need close observation and medical care by the disease specialist. The disease specialist will need to manage disease manifestations until recovery of sufficient gene-modified cells. And note that data collection is crucial during the follow-up period. Both biomarker data and quality of life information is particularly important for understanding the effect of gene therapy on rare diseases. Medical monitoring and protection of patient safety is an ongoing process throughout the complex procedure of ex vivo gene therapy. This requires solid oversight by the sponsor and CRO. We anticipate frequent adverse events. Most of the early adverse events will be related to busulfan, including leukopenia, thrombocytopenia, neutropenic fevers, stomatitis, nausea, vomiting, and abdominal pain, and serious infections may occur during the neutropenic period. Other organ toxicities are possible, but much less likely. Close monitoring after hospital discharge is also required, and we do anticipate a high rate of SAEs will occur after hospital discharge. And finally, long-term follow-up of these patients is critical to monitor for late genotoxicity and off-target effects. Therefore, depending on the vector and the type of gene therapy platform used, retention of patients in a long-term follow-up protocol will be required for 5 to 15 years. Note that there has been documented cases of acute myeloid leukemia as a late effect following some gene therapy procedures. As you've heard, ex vivo gene therapy procedures are complex, and the medical and operation challenges are many. What can we look forward to in the future to overcome these challenges? Obtaining large numbers of hematopoietic stem cells for gene modification remains a challenge. Newer mobilizing agents may shorten the collection times and/or improve stem cell fitness. Improved transduction and culture techniques during ex vivo gene manipulation may help in maintaining stem cell fitness and long-term repopulating capacity. Current protocols require specialized GMP facilities, extensive quality control, and patient-specific manufacturing, all of which contribute to time and expense. Innovations in the manufacturing process, such as self-contained bedside systems, are being developed. From a clinical perspective, there is encouraging progress in the development of non-genotoxic conditioning to reduce early toxicities related to busulfan. These progress use selective antigen antibodies to selectively target surface antigens on hematopoietic stem cells, which avoids off-target tissue damage. This potentially eliminates the risks of fertility loss, organ damage, and secondary malignancies. Antibody-based conditioning currently being explored target the CD117 or c-KIT antigen or the CD45 antigen, both expressed on the hematopoietic stem cell. The strategy for appropriateness of non-genotoxic conditioning really depends on the disease-specific requirements for engraftment. For example, most primary immunodeficiency diseases do not need high-level engraftment in order to be ameliorated. Therefore, antibody-based or low-dose busulfan conditioning is sufficient due to a selective advantage of the corrected cells. Whereas diseases such as sickle cell disease or other hemoglobinopathies currently require myeloablative conditioning because these disorders require higher levels of engraftment. Finally, the most promising development is that of in vivo gene editing. This approach could democratize access to gene therapy by eliminating the complex manufacturing, eliminating the need for conditioning, and overall reducing the costs of the procedure. To summarize, the convergence of targeted conditioning, improved ex vivo editing platforms, and emerging in vivo delivery technology promises to expand the therapeutic window for gene therapy, making these transformative treatments accessible to broader patient populations with reduced toxicity profile. As we conclude today's discussion, one theme stands out. Rare disease clinical development is rapidly evolving, and success in this space requires deep and integrated expertise. At MedPace, we stay ahead of emerging trends by combining scientific insight, operational excellence, and regulatory intelligence. Our operational teams bring hands-on experience designing and executing complex rare disease trials, including platform trials, helping sponsors navigate innovative study designs with precision and efficiency. Our medical experts remain at the forefront of emerging technology, including ex vivo gene therapies and other advanced modalities, ensuring development strategies are grounded in strong scientific and clinical understanding. At the same time, our global regulatory affairs teams closely track evolving orphan drug frameworks, providing strategic and proactive guidance. It is the integration of these capabilities and experts that allows us to make the complex seamless. By bringing together cross-functional expertise, we help sponsors confidently navigate the unique challenges of rare disease development, ultimately bringing new therapies to patients. Thank you all for your attention. And now we shift focus to the Q&A portion of the webinar, and we'll answer some questions that we received from the audience today. The first question is for Jackie. How do you structure your screening, enrollment, and patient identification across participating sites to ensure you consistently find and enroll eligible patients in multiple ISAs in the platform?
Well, that's a great question. Patient identification, as we know, is always at the heart of ensuring sites who are selected have access to the targeted patient population. However, having a highly proactive, centralized, and data-driven approach are operational tools that we have found that are absolutely necessary to carry this out. In rare diseases, patients can oftentimes be difficult to identify, and you have to build systems that continuously surface them. So one way we do this is by building a central screening hub that supports all ISAs across the platform. And this is a great way to track pre-screening efforts and potential patients who may be eligible to enroll in any of the ISAs. So keeping a close eye on when these potential patients are expected to screen, and then of course also following them closely through the screening process will also help support consistent enrollment in any of the open ISAs. Keeping track of reasons for pre-screen fails and of course screen fails also gives insight into trending at the master level as well as the ISA level. This can sometimes, of course, trigger necessary improvements and adjustments that may further enhance recruitment. So this centralized hub also continues to be a database supportive of future ISAs with accumulated lists of potential patients who may be able to be reapproached later in the platform. And again, working closely with sites with user-friendly and intuitive systems to track this information really has proven to be very successful.
Great, thank you, Jackie. The next question is for Tanya. Does RDEP replace the need for randomized controlled trial?
Thank you for your question. Not necessarily. Rare disease evidence principles provide a pathway where randomized clinical trials are logistically or ethically impossible. You must prove to the FDA during the pre-pivotal meeting why a randomized control trial cannot be conducted. And, for example, observational studies are suitable for the program.
Thank you for that informative answer. That's all the questions we have time for today. I'm sorry. We'll be sure to follow up on additional questions that we received. But now we will hand it back over to XTalks for closing remarks.
Well, thank you very much for those answers. We've reached the end of the question and answer portion of this webinar. If we couldn't attend to your questions, the team at MedPace may follow up with you, or if you have further questions, direct them to the email address on your screen. Thank you for everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us improve our webinars. Additionally, I have shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so that they may register for the recording here as well. For those watching via LinkedIn Live event, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us in thanking our speakers for their time here today. We hope you found this webinar informative. Have a great day, everyone.